Right.
Yeah.
All right, great. Welcome everyone to the next Why and How lecture. It's my pleasure to introduce Dr. Cristin Sander, who was not our original scheduled speaker. Some of you may have noticed that. We were originally supposed to have Sam Mell from the help desk present on remote computing, but he had a family scheduling issue. And fortunately, Cristin's given this talk a few times before and was able to come in last minute and give her lecture in advance. She was going to give it in November anyway. So we're very thankful that Cristin was able to do that on short notice. Also jet lag, she just got back from Spain. So today she's going to present to us an introduction on positron emission tomography, how the images are acquired, and what we could use with them. So thank you very much, Cristin.
All right, thanks Avery for the very nice introduction. And well, in the next hour, you're going to get an overview of really what it takes to do a positron emission tomography scan or study and what are the things involved. But to start out with, I wanted to just start out with an image of an example PET and an example MRI scan. If you've been coming to this series, you've seen some of the basic physics of MR images, but PET imaging is a very different type of modality, and so the images that we get look very different. This is just one example of how a PET image can look like, and what you can already see on the image is that you get very good sensitivity and specificity for a number of biological processes. PET itself is known as a quantitative technique, so we can really image molecular concentrations in the body. But there are also some limitations that PET has compared to MRI. So it has lesser spatial resolution, and there is limited anatomical information depending on what kind of tracer you use, and we're going to get into that. And as you probably all know, of course, with MRI, you get really good soft tissue contrast. You can get really good high-resolution anatomy with MRI. You get a relatively good temporal resolution when you do functional imaging, but you also have a somewhat poor sensitivity for molecular processes in MRI specifically. So just to give you this as a little visual starter, let's go back to the basics. I'm going to talk more about, or this hour, I'm going to talk about positron emission tomography. So where does this come from? A positron is really an anti-electron. It has the same charge, but a positive charge compared to the electron. Emission means really that any radioactivity is emitted from an, in this case, unstable nucleus. And tomography comes from Greek words tomos and graphia, which really means imaging a slice or section of the body in this case. So the positron emission part really comes from the fact that we use a radionuclide. And in this case, we start out with an unstable paranucleus that is produced. And there is a proton in that nucleus that decays to a neutron. And during this decay, what's given out is a neutrino, but it also what's given out is a positron. And when that positron is emitted, what happens is that this positron travels for a little bit of a distance called the positron range away from the nucleus, and it travels until it meets an electron. And when the positron and an electron meet together, they annihilate. And during this annihilation process, two 511 keV photons are produced that are given away at a 180-degree angle, at approximately 180-degree angle. And it's exactly those two 511 keV photons that are emitted during this annihilation process that we're imaging in positron emission tomography. Now, why did I start out with this? All of this entire process actually happens inside the body, because what we're doing in positron emission tomography is we inject a radioactive tracer intravenously into the bloodstream of a subject. And so this entire process that I just talked about happens inside the body, and when you have a patient placed inside a tomographic scanner that has PET detectors, we can then detect these 211 keV photons via a coincidence detection using a 3D ring of detectors. And just to give you an idea about where in the electromagnetic spectrum we're lying with these kinds of energies, I've talked about the energies that we're looking at being at 511 keV. So that's all the way to the right of the spectrum here in the gamma ray region. Whereas if you're talking about MRI, we're talking about much, much lower energy, so all the way on the left here. And in between, we have the visible light spectrum. So overall, positron emission tomography, it is an ionizing radiation that you have, but that also means that effectively any the photons that you can detect get emitted from inside the subject, but penetrates the tissue and then reaches the detector. And we're going to talk a little bit about this penetration through tissues as well, and what happens with that. So that's the very quick overview of the basic physics in PET. What I'm going to do from now on in this talk is to go over the steps of what happens during a PET imaging process. So what we do typically in any PET imaging experiment, you would have a radioactive tracer being produced by a cyclotron. The radiopharmacy produces a radiotracer, which is your isotope attached to a very specific molecule. This gets injected into the subject. You acquire data. You reconstruct the data, get your PET image, and then you would do you can do different types of analyses for your PET images. If you do quantification, you would want to apply a model, and you can get some outcome parameters such as glucose metabolism or receptor occupancy from this process. So this is a big picture overview. I'm going to return to this in this talk, but I'm going to talk about each of these facets a little bit more in detail throughout my talk now. And that brings me to the quiz number 1. So this is going to be an interactive session. I'm going to ask you questions, and there will be several quizzes throughout, and I'm going to see how much has stuck with you from a little section that I just talked about. So let's start. What have you already learned so far? And please just really shout out the answer here. So I'm going to start here. So in PET, a blank is injected intravenously. Yes, very good. Excellent. So next, the unstable radionuclide emits a... Very good. Next one. When the positron combines with an electron, this is called... Excellent. You're getting really, really good at this. All right. Next one. The annihilation gives rise to 2 blank at an 180-degree angle. Yes, I think I heard it somewhere. So it's 2 511 keV photons. And then a ring of PET detectors measures the pairs of photons using what? This is a tough one. There we go. Yes, thank you. Coincidence detection. Excellent. You've passed the first quiz. Next, we're going to talk a little bit more about the cyclotron and the production of the radiotracer. So what happens here is that, first of all, you need to produce a radioactive isotopes. The kinds of isotopes we work with in PET imaging are relatively short-lived. So they tend to be examples like here are listed at carbon-11 or F-18. And they have half-lives of 20 minutes or in the case of F-18, 2 hours. So what you have to do is to produce these kinds of tracers on site, because if you were to produce them too far away, by the time you get them delivered to the site of the scanner, you wouldn't get much signal. And so they're really short-lived traces, such as oxygen-15, that have a half-life of 2 minutes. And so it's, for any PET facility, you generally need to have a cyclotron either very close by or on site. And here at the Martinos Center, we do have a cyclotron that's downstairs next to the PET, scanners. So basically, the majority of radiotracers are produced on site and shipped directly from the radiopharmacy to the scanner bay. And so what happens is that that a proton beam bombards and your initial isotope, producing a radioactive isotope, in this case C11, using a byproduct. And all of that happens inside the cyclotron itself. I'm not going to, there's some really interesting, cool physics in terms of the cyclotrons. I'm not going to go into details in that directly. If you're interested in that, I can point you to some literature on that. But as a second step, once you have your radioactive isotope, so in this case, C11, you then need to attach your radioactive isotope to a molecule of interest. This is your biological molecule of interest that you would like to image. And in this case, there's a number of chemical steps involved, and it's really important to do purification, formulation, all within a very controlled environment so that everything stays sterile. And you have to also remember that anything that is produced during this process, some of the things can be automated, but some of them actually need a human to be involved. So they are going to be dealing during this time with radioactivity. And so you want to be producing your final tracer as soon as possible, or as fast as possible, to minimize radiation exposure. But once that's done, you can then start to image and inject the radiotracer into the patient. And I think it's important to point out this entire process, because you can see that the isotope production can take on the order of 40-45 minutes, depending on the isotope. And then it can take something on the order of an hour, it can be shorter or longer, for the actual radiotracer to be produced. And so if you have a scan scheduled at 3 p.m. in the afternoon, the radiochemistry team will start working at maybe noon to get your radiotracer going, in order so that you can have the radiotracer injected at the proper time. So timing is really, really important in PET imaging. So let's talk a little bit about units of radioactivity. I've talked a lot about radioactive tracers, but how do we quantify that? And really there are two types of units that I use, but the SI unit of radioactivity is becquerel. And 1 becquerel really means that you have 1 decay per second. In the US, we actually commonly use radioactivity in units of curies. And it's really equivalent to becquerels, where 1 nanocurie is equivalent to 37 becquerels. So it's a linear conversion. It's just traditionally in the US, we tend to talk about curies or millicuries as the radioactivity unit. And I've already talked about the fact that time is really essential in PET. But this is how a typical radioactive decay curve can look like. And this is for the example of an isotope F18 or C11. And when I talk about a half-life of an isotope, this really means that it's the time it takes for half of the atoms in your sample to decay. So if you start out with, really what that means is that effectively a short half-life means that you have a less stable radionuclide, and this decay always follows an exponential decay. So as an example, if you start out with 5 millicuries of C11 tracer and you wait 20 minutes, you're going to lose half of that radioactivity during that 20 minutes. So after 20 minutes, you're going to be left with 2.5 millicuries. And that's directly proportional to how much signal you can image with your PET camera. So time is really, really important. And knowing what the half-life of your isotope is that you're imaging is also really important to calculate that. So I've talked about the chemistry of the radiotracer, but let's talk about the biology a little bit, because it's really about asking what kind of targets can you image in the human body if you want to do PET imaging. And in general, when you're starting out with a new PET study, I would encourage you to think about it like this, not necessarily, oh, we have this tracer, now we can look at something, but ask about what's the target of interest, and then look about what kind of tools we have to image that. And broadly speaking, there are two categories of PET tracers. One of them is receptor or ligand based. That means that the radiotracer fits into a receptor ligand, just like a key fits into a lock. And you can image that there in this case, and one of the examples is to look at dopamine receptors that you can image where your radiotracer is a molecule that specifically fits to that receptor and gives you a very specific signal to just that specific receptor. Another type of radiotracer is really enzyme or substrate based. That means that the radiotracer really interacts, for example, with an enzyme and creates a byproduct from that. And in some cases, that gets trapped in a cell. That's really the case what happens with FDG, which looks at glucose metabolism. Well, those are broad categories of PET tracers. There are a number of really different subcategories within that. But since I've talked about FDG, if you've heard about PET, that's probably the first tracer you have heard of it. You've heard of it's fluorodeoxyglucose, and it measures glucose metabolism. Clinically, that is going to be 95% of your PET scans. It is a gold standard to use FDG PET for oncologic imaging because tumor cells really divide at a much higher rate, so they use much more glucose compared to other tissue cells in your body. So it really comes out as this bright spot and has very, very good contrast compared to other tissue types. That's why it's the gold standard in cancer imaging. And this is just an example of a map in the US of the distribution of the FDG network. But in addition to imaging cancers, FDG can also really look at glucose utilization. And being at the Martinos Center, of course, we're interested in brain imaging applications. And just keeping in mind here that even though the brain represents only 2% of the body weight, it actually uses 25% of your total glucose. And in FDG, what you're looking at is really so this on the left is your FDG tracer. It crosses the blood-brain barrier and then interacts with this enzyme hexokinase. And when it's modified, it becomes trapped in the cell and is really metabolically trapped. And so that's how you can measure an increase in glucose utilization because the chemical structure has changed due to the interaction with the enzyme. And so when you are talking about brain mapping, which some of you might be doing, one of the very first brain mapping studies was actually done with FDG PET, not with fMRI. And here you can see an example from back in the 80s where a visual stimulus showed an increase using FDG PET in the occipital cortex. So FDG has many types of different uses if you're thinking about your the kind of question you're asking. So a little bit more details about receptor radiotracers, and this is specifically about in vivo imaging with receptor tracer. And what I want to show you here is that we always say that we're injecting a radiotracer. Now, why do we say that? The radiotracer means that we're injecting, or the tracer part in radiotracer means that we are injecting a very, very small amount of this molecule. And so what we get is that the PET signal is proportional to density of whatever target we're imaging. But the key point here is because we are injecting a tracer, we're not disturbing biology. We're not saturating any kinds of receptors with our PET radiotracer. And so in general, what that allows us to do is if you have a high density target, so you have many receptors within a given region, you would expect your radiotracer to bind proportionally to larger amounts because there's more receptors. Whereas if you have a low density target, you will get a lower signal because you have fewer concentrations of receptors in that target. And that's how you can distinguish concentrations. But in vivo, it's also, there's a caveat to this, because what you can have is you can also have endogenous ligands. So especially if you're talking about a neuroreceptor, you will have endogenous neurotransmitters. So if let's say, for example, there's the dopamine receptor you're looking at, you will have endogenous dopamine, so a neurotransmitter that's abundantly available in the brain. So one of the things that PET is can actually not do is distinguish in this case between the top and the bottom targets, because now what you have is you might have a high density target, but if some of those receptors are occupied by an endogenous ligand, to the PET tracer, it will still only seem that there are so many number of available receptors there as in the low density target. So what we're really looking at with PET imaging is the number of available receptors. Not always truly about receptor concentrations, but the total number of available receptors. So it's just a detail to keep in mind that's important in certain cases. But overall, there's a lot of information that PET can provide. One, we can learn about the pharmacokinetics. This is really the information about the radiolabeled drug itself. We can also learn about pharmacodynamics, because this is now the effects the body has on a drug and gives you information about a biological event inside the body. And we really get both of these combined together, because a typical curve in PET imaging looks something like this, where you get a mixture of your pharmacokinetics from the injection of the tracer, but you also get information about how the body is affected and how that tracer affects with biological events in the body. And there's a little note here that I have, is that the uniqueness of the radiotracer is that they have saturable binding. So that means that a good radiotracer, if you inject it into the body, you will want to see a specific signal. That means that if you were to give a pharmacological blocking dose of that same drug with that tracer, you would see the signal go down significantly. And that's known as saturable binding of the radiotracer. And I just wanted to give you a couple of examples of what radiotracers are. This is an example of the dopamine system. And you can see here the presynaptic and the postsynaptic membrane. And there are actually several targets of radiotracers that you can look at. So you can look at a dopamine transporter that's sitting presynaptically that transports dopamine in and out of the cell depending on demand. There's the actual postsynaptic tracers that are important for signaling down in your postsynaptic axon. You have a specific molecule, a PET tracer that can look at that. You can look at surrounding glial cells using monoamine oxidase tracers, or really look at overall metabolism using FDG glucose. So within that same synapse, you can look at different kinds of molecular events with different radiotracers. So again, depending on the kind of question you have, there might be different kinds of real suitable tracers available. And just to go through a couple of examples, a C11 raclopride is a radiotracer that's been very well validated and looks at the dopamine receptor subtype D2 and D3. It's been used in a number of different kinds of imaging studies, especially in psychiatry. And there's specific agents for the mu-opioid receptor system, looking at, for example, now the opioid crisis we have. We can specifically image the opioid receptors in the brain or in the rest of the body. And one of the tracers available for this is C11 parfentanil. And another example, this is actually looking at amyloid deposition now. There's actually a lot of newer tracers that also look at tau. This is really important in aging and in Alzheimer's disease. You know, if you're familiar with it, you will know that we know that there are depositions of these aggregated proteins in the brain, and we can image them with specific PET ligands. And again, I just wanted to give you a couple of examples, but if you want to really get a big spectrum of this, I would really refer you to this website from the National Institute of Mental Health. They have an updated list of overall brain targets and really currently existing radiotracers. So currently, as of today, there's 39 separate brain targets that you can image with PET ligands, and there are more than 100 ligands available for that. Sometimes a specific target has several tracers with different properties to look at. But if you're looking for a specific target in your study, go to this website and just browse and see what you can do with different kinds of radiotracers. So that brings me to quiz number 2. So radioisotopes are produced by a, yes, very good, by a cyclotron. Units of radioactivity are blank and blank. Yes, I hear a lot of murmuring here. Feel free to speak up, but I did hear it. Becquerel and Curie or millicurie. Then radiotracers are most commonly labeled with blank or blank. Yes, and yes, exactly. C11 and F18. Those are the most commonly most common radioisotopes. All right. The time it takes for 12 millicuries of F18 to decay to 6 millicuries is called. Yes, you got this one. And what's the half-life for F18? Oh, I heard some rights and some wrongs. It's yes, it's about 2 hours. C11 is 20 minutes. So F18 FDG can measure what kind of metabolism? Excellent, very good. Quiz number 2 is done. So now we move on to tracer injection and PET scanning. So with that, I'm gonna switch to a brief video that kind of summarizes what we've actually looked at so far. And it'll give you a little overview of what happens with the radiotracer. Let me try to go here. There we go. Oh. All right. I'm gonna restart this. There we go. So what you see here is that you have a radiotracer that's injected into the bloodstream. And so once it's injected, it takes a little bit of time to distribute throughout the entire body. And when you then wait, again, this is about imaging biological processes. In the case of FDG, for example, it goes to organs that are very high in glucose metabolism, and that's how the contrast in your PET image is created. In this case, you can look at really tumor tissue. And so this is going back to your bloodstream where your radiotracer really distributes into, it diffuses out of your bloodstream into the tissue. The F18 decays, it meets the electron and annihilates, giving away 2 511 keV photons. And that's what we're detecting in the PET scanner, right? And then magically the image appears on the screen. So let's talk about how we get from no, that's not what I want to do. I want to go back to my PowerPoint. Maybe I need to get out of full screen. There we go. And here I am. Okay. So let's talk about how we get from the actual acquisition to an image on the screen. And here I just want to point out to you that there are different kinds of sensitivities in PET. And a little bit of a different way of how we think about sensitivity in PET. So on the left, you can see based on the image, you have a very highly sensitive scan. On the right, you have low sensitivity, you have a lot of noise. But you can achieve high sensitivity by either scanning for a very, very long time. So if you're scanning for 30 minutes versus 10 minutes, you're gonna get much more sensitivity. But you can also get higher sensitivity if you inject more radioactivity into your patient. So if you inject 3 times the radioactivity, you can scan for shorter and still get the same good quality image out. Of course, there's a trade-off here with how much radioactivity you want to expose your patient to. So it's just important to keep those things in mind because they're different from other imaging modalities where you don't inject your tracer. And of course, sensitivity is affected by by your PET detector, the number of detectors you have, and the actual detector makeup itself. But importantly, the way we detect events or photons in PET is really through 3 types of events. First, there are true coincidences, which are annihilation photons that just escape the body without any interaction with the surrounding tissue. And that's really what we're interested in, right? That's your perfect case scenario. But of course, it doesn't always work just like that. What we also detect is random coincidences. And a random coincidence is, it happens when you have 2 annihilation photons from 2 separate emissions in the body that happen to strike the detectors at the same time. And so these random coincidences falsely get attributed to a different original event, as you can see in that connecting line here. And finally, there are also scatter coincidences, in which case you have 2 photons that get emitted, but one or both of them are scattered a little bit because they interact with tissue. And so the true line of response is a little bit altered. And again, this leads to events that are being wrongly attributed to a wrong position in the body. So both these random and scatter coincidences occur because you have attenuation of photons in PET imaging. Photons of 511 keV are very high energy, but they get attenuated if they interact with matter. And it leads to an increase in image noise, really. And what we have to do in PET imaging is to correct for these kinds of attenuations. So both for scatter, we correct for scatter, we correct for the randoms as well. But overall, we also correct for overall attenuation. So on the left here, you can see an example of just a phantom, a cylinder that has uniform activity. And if you don't correct for attenuation, you will get an image that looks like on the left, where you have a really bright rim on the outside, because photons that are on the outside, they don't have to pass through a lot of matter. And so the likelihood that they get attenuated is much less. And so you get this hot ring at the edge that goes, becomes smaller as you go to the inside. When you then correct for attenuation, you can get your reconstruction of the uniform cylinder. And how do you do that? Well, overall, it's you can implement it conceptually very straightforward, where your attenuation correction is really dependent on two factors. It's the total distance that photon crosses through your object of interest, and it's the distance from the original point of interest to the outside of that. So it's really the distance that it crosses entirely through the phantom, or sorry, so the sentinel subject, whatever you're imaging, really. And the other factor it depends on is the attenuation coefficient, mu that we call it. And this mu really depends on the atomic weight of the material, because if you have a very dense material, the likelihood of interaction with that matter is much higher. So for example, bone is highly attenuating compared to soft tissue, which is less attenuating. And so we need to account for this in attenuation corrections. There are a number of other data corrections that need to be done. And I'm not going to go into details, but if you are really interested or working in PET image reconstruction, these are the kinds of things like normalization, dead time pileup, that really need to be accounted for during the image reconstruction process. But let me talk a little bit about and give you an overview of the actual reconstruction process. The raw data space that we acquire data in is called the sinogram. And the sinogram is really a space of an angle versus a radius. What that means is that if we have a photon that escapes, in this case, the brain, from the center of the brain, you would start out, or you really would collect all the lines of responses from the center out for every angle. So what that means is that for each radius, you get really, for each specific radius and angle, you get 1 point in your sinogram, right? So that really defines a point in your sinogram. And if you then acquire all the same lines of response at the same angle, you will get one line in your sinogram at that angle for all the radii that you can acquire. If you then change your, look at a different angle, you will get a different line in your sinogram. And so overall, a sinogram looks something like this, where you have a unique pairing of radius and angles as they come in through the PET detection, but that's how it's really recorded in the raw data of your PET scanner. So once you have your sinogram, you then need to reconstruct this in order to actually receive an image that's biologically relevant. And for the sake of completeness, I'm just going to mention filtered backprojection. This is how traditionally images were reconstructed. Nowadays, it's usually done with an iterative algorithm, but really conceptually speaking, it's filtered backprojection is easy to just follow. So when you have an image, you can project it for different angles. And if you project it for one angle or for several angles, you can then really get your image reconstructed through a filtered backprojection. And that can be done through many iterations until you go from your original data acquisition to your actual image. Like I said, this is typically not done on newer scanners anymore. On newer scanners, we really do reconstruction through an iterative reconstruction process using expectation maximization algorithms. So here what you have is you would start out with you would just project the image to get an initial estimate of an image, and then you project it back to sinogram, you compare it with your actual sinogram, you compute the error, and then you re-project the image in order to get an update of that image. And so going through this several times, you can get from your estimated projection to your actual real projection with an estimated image that aligns with your actual measured projection. So that brings me to quiz number 3. All right, this one is on image reconstruction. So events detected in PET can be classified into 3 types of events. What are they? Yes, trues, randoms, and scatter. Excellent, you are excellent. Attenuation of photons traveling within an object depends on the total distance in the object, and what else? Yes, thank you very much. Attenuation coefficient. The raw image data in PET is stored, oh, sorry, there we go. Raw image data in PET is stored in a sinogram. That's correct. That's the raw image space. And then each blank in the sinogram corresponds to coincidences observed for 1 angle and 1 radius. Yes, each point. And then image reconstruction can be achieved by 2 methods. What are they?
Direct reconstruction and iterative reconstruction.
Yes, so it's filtered back projection or iterative reconstruction. Great, you've passed quiz number 3. So let's go on to what do we do with our PET image data when we have it in hand. And so what we can do in PET, I mentioned this at the very beginning, is that we can quantify PET images to look at really molecular concentrations. In order to do that, we need to do kinetic modeling. Why do we need to do that? So this is really the justification for it. So when you have a PET image, as you see here, when you're looking at a voxel, you have, you're really looking at whatever, or really what you're interested in is the radioligand that's bound to your target of interest, right? But when you're looking at a PET image, you don't only have, you're not looking at purely just your specific binding of that tracer to your target. You also see that there is some radioligand that can be floating in free tissue because, right, we're injecting it into the bloodstream and it diffuses into free tissue. So not all of your radiotracer is sitting at that ideal target. So that's also in your voxel. What's also, in addition to that in your voxel, is maybe some blood vessels where radioligand is still passing through. It might be diffusing in and diffusing out, but there might still be residual radioligand in blood plasma that can change over the course of your PET scan. So you have all of these really what we call compartments in one voxel of your image. And because we have these different types of radiotracer bindings or really the existence of radiotracers in different types of tissues, we come up with compartmental models to quantify that. And so this is really an example of a model where you can express the concentration of radiotracer you have in plasma, the concentration you have in non-displaceable or free tissue, like free floating in tissue. And then you, of course, have the specifically bound ligand that you want to really measure. And this is a typical time activity curve, so the PET signal over time for a non-specific binding and specific binding time activity curve from that are corresponding to these compartments, and just to give you an idea of how they can vary in time. And when we have these compartmental models, the way we describe them is that you can then use the law of mass action in order to describe them as first order differential equations. Again, I'm not going to go into details of this, but I just want you to understand that that's how we mathematically describe a compartmental model. That's in this case the example above. And right now, all you need to know is the fact that your CP, or your plasma concentration, is generally used as an input. So in the ideal world, you know what your plasma concentration is like, and that you can actually measure that in a patient when taking blood samples. And the other known that you have here is your PET signal. So it's you acquired your imaging signal for each voxel, right? And so the PET signal is both the non-displaceable and the specific binding tissue combined. And if you image late in the scan, you won't necessarily have too much overlap with your blood volume, but it's a little bit tracer dependent. And we know those parameters, and what we're interested in is actually to estimate these rate constants. So it's K1, K2 through K4, and different radiotracers have different models because they do describe the biological events that radiotracer goes through. But you would want to solve for these kinetic rate constants, giving the knowns that you have. And it's possible to do that with these some differential equations. But there are many different types of models that you can apply in this context. So overall, I just want you to be aware that they are both microparameters and macroparameters. Macroparameters are really individual rate constants that I just pointed out that really are theoretically defined well and that we can estimate. But it's really hard because there's a lot of variability, because it's really a we have too many unknowns for the number of knowns that we have in just this simple equation. So what we do is we rearrange some of the equations in order to compute compound variables. And so they are usually composed of ratios of some of these microparameters and kinetic parameters, but they do have biological meaning. And they are much more stable as a final outcome measure to quantify. And so what examples of this is binding potential or volume of distribution, that is an outcome parameter in PET. So binding potential, as one example, for a two-tissue compartment, it really tells you about the specifically bound ligand. It's defined as the equilibrium ratio of specific binding relative to a reference concentration. And this reference can either be, for example, a type of tissue that doesn't have any specific binding. Some radiotracers and some radiotracers, you can have this delineated by anatomical information, because you know that certain parts in the body or in the brain don't bind that target. So you can use that as your reference, or you can use a blood concentration as your reference for your binding potential. And so it's really the relative, it's really that ratio that defines binding potential. And if you're looking at changes in your signal, you can have a baseline binding potential, and then your relative change in binding potential is something where you can then talk about changes in your target occupancy. So that brings me to quiz number 4. And this is my final quiz on kinetic modeling. So here we have an example of a kinetic model with 3 compartments that you can see here. And what, well, I actually gave this one away. This is a compartment. But I'm not going to give the next one away. What are these parameters called? Yes, very good. Kinetic rate constants. And what's this red compartment here? Yes, it's a plasma concentration, or also known as your arterial input function. What about the orange one? Yes, I heard it. It's either the free or non-displaceable ligand in tissue. And then the blue one? Yes, it's specifically bound ligand. And then what's this dotted line here? If you take both the non-displaceable and specifically bound ligand together. That's the tissue concentration. That's the tracer that's in tissue versus blood. Those are two just separate compartments again. And then overall, that green box, what is that? What are we measuring overall? Yes, there we go. It's so simple. You don't even think about it. It's your PET signal. And then what's an example of a macroparameter? Yes, binding potential. And then what kind of outcomes can we calculate from binding potential? I think I heard a whisper somewhere. Yes, it's occupancy. It's receptor occupancy from binding potential, from changes in binding potential. All right, excellent. You are amazing at this late hour of the day. So I just wanted to point out a couple of tools that are available to do kinetic modeling. There is one commercial tool out there that's PMOD that can really take your PET signal, and you can have a choice of different kinds of kinetic models. There's an in-house software that we also develop, and it's developed by Joe Mandeville, that looks at really dynamic changes in binding potential. There's also the London group that has developed MeerKAT, and that's a freeware. So there are different options. I would say a lot of PET imaging centers have their in-house pipeline. There's not one software that's established. At this point, I would say PMOD is probably most widely distributed overall in terms of imaging centers, but it really depends on what kind of study you're doing for what software is appropriate. All right, and I know we're almost up in time, so I just wanted to really quickly give you a couple of slides on combined PET-MR imaging, because here at the Martinos Center, we have combined PET and MR imaging. This is really a scanner that was introduced about a decade ago now, but is still a relatively new technology, especially in the clinical realm. And here at the Martinos Center, we have two human-sized PET-MR scanners, that's in Bay 6 and in Bay 7. And you can truly simultaneously acquire both your MR and your PET images. One is for the brain only, which is a high-resolution BrainPET, and the other one is a commercially available Siemens Biograph MMR that allows you to do whole-body combined PET-MR imaging. And PET-MRI is really an active research field. There's a number of different kinds of applications that are really interesting to explore with PET-MR. I think one of the things that have transpired over time is the fact that you can use your MR over time to do motion correction of your PET images. You can look at using a non-invasive arterial input function, which really reduces burden on taking blood samples from the patient itself, which can sometimes painful and also very cumbersome, and it's not necessarily something that you want to do routinely clinical. Compared to PET-CT, which is how standardly PET imaging is done, you minimize radiation exposure significantly because taking away the CT for doing attenuation correction, you can now replace it with non-ionizing radiation from your MRI. And there are some areas of the body where also CT anatomy is suboptimal. We are very good at imaging soft tissue with MRI, and so there are certain specific applications that are also very interesting for that. Some of the things that we do in our lab is to look at really truly simultaneous dynamic signals where you can really use the simultaneity of the scanner and track both dynamic molecular events over time together with functional MRI markers. And so this is just one example of the neurovascular coupling to PET and fMRI images. And so with that, I'd like to thank you for being here and listening to this late hour talk. I think hopefully you've learned that it takes a big team to do PET imaging. You have to talk to your radiochemistry. You want to understand a little bit about that PET instrumentation and reconstruction. And then finally, you want to understand how you can actually do your image analysis and kinetic modeling. And if you're interested in these kinds of studies, there are many people here at the Martinos Center that you can talk to for different parts of this, get some advice, and collaborate on that. And so finally, just consider what are the targets you're interested in your study. And of course, scan cost is also an issue where you're just, and compared to MRI, PET adds on a significant component because it takes a lot of, a big team to get PET scanning going with the radiochemistry. So thank you very much.